Welcome everyone. We are now live in one more of our seminar series as part of the worldwide series initiative. And this is a track that is run by Open Neuroscience, an open source community-led initiative that is trying to put together resources on open source tools for neurosciences. Today we have the pleasure of hosting Dr. Seb James. So Seb is going to talk to us about Stalefish, a software package that allows 2D histological data to be analyzed and also converted into 3D space and more interesting things that he's going to talk about, so I don't need to talk about it myself. Before we start, I would like to give a brief intro on Seb's work. And so he received his PhD in 2001 in experimental physics at the University of Cambridge. And then he worked in a number of software engineering jobs, developing his coding skills and eventually becoming a partner at WML, which is a software business with customers in the UK and abroad. And in 2013, when he completed the development of this company's software product, he returned to academia. And so this is, I guess, an interesting talk on itself, like how it is to go to industry and then back to academia. But he joined Kevin Kearney's research group at the University of Sheffield in the Department of Psychology as a postdoctoral researcher. He's now working with Stuart Wilson and is interested in the developmental biology of the brain. And he's using computational modeling techniques to explore the mechanisms by which the brain wires itself up. Seb, thank you very much once again from me and from all the people organizing Open Neuroscience. The floor is yours.
Great, thanks. Thanks, Andre. Thanks for the introduction. So I'll just hope that the screen sharing works. Is that okay? Can we see that?
Yeah.
Okay, and if I do this sort of presentation mode, yeah, that's working. Brilliant. Okay, so I'm going to talk about this this tool that we've developed called Stalefish, and it's all about getting more from your 2D histology slices. Okay, so histology is a mechanism by which you can study animal and plant tissue, and you slice the tissue up and stain it to reveal information about the tissue which you couldn't otherwise see. Most of the slides that I'll show are all in-situ hybridization studies. So the staining is revealing a particular gene. In most cases, it's a gene called ID2. So this picture in the middle here is a 2D slice of the brain on the left, which is a vole brain. The slice has been taken out of one of the hemispheres of the brain, about halfway down it. And the areas of the slice where the stain is sort of dark, tending towards black, those are the regions where you have large numbers of messenger RNA molecules that code for the gene ID2. And what you can see in this slice is that the cortex of the vole's brain, which is this area around sort of the top, has got this beautiful layer-based expression of the gene. So you can sort of see superficial layers and then there's central layers and there's lots of structure there. And then you can also see expression in the spiral of the hippocampus here. Okay, so we're talking about histology. And this story starts in the lab of Leah Krubitzer, who's pictured on the right there. Her lab is in California at UC Davis. And she has a PhD student called Mackenzie, Englund. And Mackenzie was thinking about how to look at gene expression across the cortex. He wanted to quantify cortex-wide gene expression, and he also was interested in layer-specific expression. And one thing that the Krubitzer lab is well known for is looking at lots of interesting species. So Leah has done work on duck-billed platypuses. She's done work on dolphins, all sorts of species. They do a lot of work with wild rats, so they're interested in animals that are behaving naturally. So they don't restrict themselves to the sort of lab mouse, lab rat kind of paradigm. They're interested in all sorts of animals. And so they're interested in comparing gene expression across species and across ages and experimental conditions. So if we're thinking about expression across the cortex, actually, let's just move on to, I'll just talk about the collaboration before I get into the science. We come into this, or I come into this story because I'm a member of Stuart Wilson's lab at the University of Sheffield. And Stuart is on a grant with Leah, along with Kelly Huffman. So Kelly's pictured here on the right. This is Stuart on the left, and they're all together on this McDonald grant called Cortical Plasticity Within and Across Lifetimes. Okay, so we are the computational people, and we've been thinking about how patterns of gene expression can form and how we could simulate those. And then Kelly's lab does lots and lots of ISH work and produces lots of experimental data, along with Leah's lab. Okay, so if you're interested in areal expression across the cortex, how do you go from those brain slices that I showed in the first slide through to areal expression? Well, one way to do it is you collect together all of the brain slices for one brain, and you analyze them very carefully, and you basically draw cartoon diagrams of the expression based on studying the slices. It's not particularly quantitative, and it takes a lot of sort of time and expertise to do this, but this is a method that people use. So this image here, this figure here, is from a paper from Kelly's lab from a few years ago, and it applies just this technique, looking at different areal expression of a number of genes in mouse. An alternative is to, rather than slicing the brain up to do your histology, you can use the whole mount technique. You can stain the entire brain and then just take photographs of the external surface of the brain, which is seen here for a gene called LMO4 in the bottom left here, and for EMX2 in the top right here. Or you can sort of take the cortex off, flatten it out, and stain the cortex, which is shown here in the top left here, where you can see the whisker barrel field nicely in that image there. The trouble with this is it loses layer-specific information, and also you're sort of throwing away some of the three-dimensional information, because by taking a photograph of a 3D brain, you're projecting that three-dimensional information onto a 2D image. Other people have thought about three-dimensional gene expression and examining three-dimensional gene expression. If you go and have a look at the Allen Developing Mouse Brain Atlas, you can find brain slice images for thousands of different genes at multiple different developmental time points in the mouse. They went through they built a very sophisticated and expensive pipeline to do this. And one of the things that they had to pay attention to was ensuring that they could register the slices with respect to each other, so they were all lined up. And by doing that, they were able to then produce three-dimensional sort of clouds of expression data. So this is an amazing library of data, but it's fixed in time. And also, if you're going to analyze the three-dimensional expression data from the Allen data set, you have to sort of post-process it. You have to decide exactly, how are you going to, are you just going to look at it and say, okay, this is it, or are you going to turn it into surfaces or maps in some way? And if you're going to do that, then you have to apply some sort of analysis to the data in any case. Okay, so that's the Allen data. And in some sense, the technique I'm going to describe to you is an alternative to what the Allen Developing Mouse Brain people did, but it's on a kind of different scale. It's on a scale which should be accessible to any neuroscience lab that's working with histology slices. Okay, so Mackenzie had a bright idea. This is a couple of years ago. when he was thinking about this problem, and his idea was that you could draw a curve around the outside of the cortex. You could draw a polynomial curve, and then if you drew if you sampled along that curve, underneath that curve at evenly spaced locations, you could get a sort of linear map of the expression profile across the cortex. And by changing how deep you sample from the curve, you could access different layers of the expression. So he set this up and did a Python implementation of this idea, and he presented that at SFN in 2019. And he called it Stalefish because here's the sort of the explanation for the name, which is slightly unusual. Stalefish, it's a snowboarding move which Mackenzie can do. He is the only member of our team who can do it, I believe. It involves doing a big jump and grabbing the board sort of mid-air. But the technique is spatial alignment of ISH data. So we think that the technique has grown beyond that description to some extent, and certainly it's not in any way limited to ISH data. Any kind of histology data where you've got stained images with structure in is accessible with this technique. Okay, so that's Mac's idea. That was the first implementation. And then Stuart was talking to Mac about it, and he liked the idea. And so because he'd been working with a library called OpenCV, which is great for opening graphics and dealing with image data, he hacked together very quickly a version of Mackenzie's program, only with a kind of more graphical interface. So this is what it looked like. You would open your brain slice image. You would then click around the structure of interest, producing these blue points. And then the program would fit a polynomial to those points, the best fit polynomial, which is the green line. And then using the normals to the polynomial line fit, if you drop those down, those would define sampling boxes. And then you could essentially sample the average luminance within each sample box to give you a data point that you could then arrange around that curve. So there's the idea again. In this case, this is a third order polynomial, I set it to 12 bins, so you can sort of say they're very big bins. You might well have more of those in real life. Okay, so there are limitations to this method, though. If you try and extend the blue data points to make the curve perhaps more complicated, to extend, this is now trying to capture the information around the whole of the cortex there. Now the third order polynomial can't sort of faithfully reproduce that shape. And so now we're not really getting exactly what we're expecting here. We're not following the curve. One solution might be to increase the order of the polynomial to allow it to fit that curve, but it turns out that that's pretty unreliable. You end up with overfitting, where you end up with a very wobbly fit. As you can see, the green line's very wobbly here, and our sampling bins aren't very useful. So we felt that the technique needed some thought. So this is where I kind of came into the story. I'd been working with Bezier curves to produce simulations of the whisker barrel field. And what I was doing is I was tracing around the barrel field in Inkscape, and then I had software which would load the curves of that barrel field, and then allow me to define an arbitrary boundary for a reaction diffusion style system, which is playing out here on this little video, which is arranging the whisker barrel field under the action of a set of gene expression gradients. Okay, so I've been working with Bezier curves. Let's see if I can move on to the next one. Yeah, a Bezier curve is just a polynomial curve. This is an example of what they look like in a drawing program. And they have these control points. So there's a control point at the start and the end of the curve, and there's one midway, and then these kind of handles that allow you to control the shape of the curve. And the nice thing about these for drawing programs is that if you arrange several Bezier curves and join them up, you can make a shape which is arbitrarily complicated. You can make any shape you like. So we thought these would be great for the curve fitting in Stalefish. And so that's what I put together. So this is an example where the user has now placed these 4 blue points to produce one Bezier curve, and then these 4 red points define the next curve. And so we've got a series of Bezier curves that are joined together to follow that curve round. And then the actual curve is the green line, and there's a little bit of code in the background which is dealing with the joining point. So it's very important that the gradient of the curve is continuous through the join. So it handles that, and you end up with this nice smooth green line, and the normals to the green line again define your sampling boxes. And once you've got the sampling boxes like that, you can collect the average luminance of the pixels in each box, and you can plot that as a function of the distance along the curve. And that's what I've done here. I've used a Python script to extract the data from Stalefish and plot it as a function of distance.
Seb, sorry, quick question.
Yeah.
Is there any reason why you're using multiple of these Bezier curves and not just one that has many data points?
Yeah, so the Bezier curve is defined. Well, if you used the Bezier curve is a third order polynomial, so it suffers from the same problem that a normal third order polynomial suffers from, which is that it can't be that complicated. So again, you can go to a higher order Bezier curve, but you get the same problems of wobbly lines. So that's why you need to fit them together. You need multiple Bezier curves. It's that issue of overfitting or getting just the right fit. So that's why it's multiple. And the way these Bezier curves are defined, so instead of having those control handles that you move around like in a drawing program, it's set up so that depending on the number of points that you define for each kind of subsection of the curve, that will define the order of the Bezier curve. So if I were to draw in 5 blue points for a Bezier curve for the first one, then I'd get a 4th order curve instead of a 3rd order. Okay. Okay, so this technique, now we can start thinking about layer specific analyses, because we can control the size of those sampling boxes. We can make them deeper, like in panel B. We can shift them away from the curve that we defined, like in panel C. We can even extend them either side of the curve. So that allows us to access different parts of the tissue. Another way to do layer specific analysis is to simply take, on the same brain slice, draw 3 different curves. So as an example, I've done that on the superficial layer of expression in this slice, on a medial layer, and on a deep layer of expression, and then I've plotted the same kind of plot for each one. So you can compare layers pretty easily now. You can take that curve and you can plot it in 3-dimensional space. So this is the same data, but now I'm plotting it as a kind of ribbon. And the amount of red saturation gives the signal value. And when you've got 3D ribbons, you can then start to take multiple slices and start to arrange those 3D ribbons in a row and start to build up a 3D surface. But then that brings up the question of how to align the ribbons. How do they arrange themselves? And that's not entirely an easy problem to solve. So I kind of came up with 2 possible methods to do this. So they're called auto align and landmark align. I'm mentioning that because that's in the documentation. So the auto align method, you basically take 2 adjacent curves. and you apply a rotation and a translation to one of the curves to bring it so that it is as close as possible to its neighbor curve. And by as close as possible, what I do is I take 100 points around each of the curves, and I just compute the sum of the squared errors between the positions of those. And so that value is minimized, and the optimal rotation and translation is found to give you that, to minimize that error. And so that's applied to each of the slices throughout the data set, and that arranges the curves fairly well. But the landmark line method is much better. You can do a lot better than that if you prepare your sample with a needle. So what you do is you mount the brain like this in, this is going to be set in a kind of gelatin albumin fixing solution. And you put a needle through sort of above the brain, so that when the fixing solution solidifies, you pull the needle out, prepare the slices as normal, and you end up with these beautiful circular marks in the slices. We using the circumcircle, we can find the center of the circle, and then that defines a straight line that we can line the slices up with. So if you do that, then you only need a 1 degree of freedom optimization to align the slices. We haven't yet tried putting 2 needles through. When I suggested this technique, the experimentalist, it was Riley in Kelly Huffman's lab, he went ahead and did this, but I wasn't sure how complicated it would be to do 2 needles. So I let him do 1, and we found that actually it worked really well. So this is how it kind of looks if you do that. The pink spheres here are the positions of the alignment marks. This is a slice set of about 50 slices, and it actually lines up really nicely and looks very much like a brain hemisphere. The white rod there is a user-defined brain axis, which is important for a later technique. This is the same data auto-aligned for comparison, and you can sort of see that the alignment is in the middle of the brain, but it goes a little bit wonky at the end, so it's not as good as the landmark alignment. But if you're analyzing existing data sets, then this may be sort of the best alignment method. Actually, there's one other alignment method that we've come up with, which is it's almost like a sort of simulated needle mark. You use your anatomical knowledge to place a landmark on each brain slice, and then it lines those up on a straight line, and that works reasonably well as well. This is really just to show off my OpenGL software code. This is the GL part of this program is all coded from scratch in C++. The ribbons are made up of little rectangles. If we take the same data points and we join them up as circles, then we can end up with a nice smooth 3-dimensional plot, and that's what I'm going to show you here. And now we start to see some structure in the expression. So this is ID2 gene expression in a vole brain, and we see sort of there's a ridge of high expression, this part, sort of the medial part of the brain. And then we see this bit here is actually kind of corresponds to the visual cortex. And there's even there's a bar of expression, which you can just about see here, which separates the visual cortex from other brain areas in the brain. Okay, so there's our plot. So for most of our simulation work, we're interested in 2-dimensional maps of gene expression. So in a way, the 3-dimensional reconstruction is just a way to get to a 2-dimensional map. And this is how we've been doing that. What we do is we take the white brain axis, which you can see here, and well, it's in all of them, and we define a row of center positions on the curves, And each center position is a fixed angle around the brain axis. And having defined that row of center positions, we then straighten out the ribbons. We flip the thing over. That's only because it then matches up with the two-dimensional image. And to turn it into a 2D map, all we do is we line up those center positions in a straight line, and that brings all the rows into alignment and gives us a 2D map. And then the last step is to resample the 2D map, which the inset here shows that the sort of the pixels of this map are rectangles, and they are rectangles of a variety of size. The actual size of the rectangle is different on each brain slice because the brain slices are of different lengths, but they all have the same number of sample boxes in them. So we resample the map so that it becomes a regular image with square pixels, and that's what we see on the right here. And having a regular image is great because you can just apply, you can load this data in Python or whatever you want and apply image processing code to it. And so now we can make maps and we can compare layers. So this is just a kind of a showcase of, this is one vol brain, and this is showing the ID2 expression in the superficial layers sort of 2/3. The layer 5 in the middle, and then the layer 6 expression at the bottom here. And we see there's quite distinct patterns of expression in the different layers. We can really see there's almost like a sort of inverse of the expression in layer 2/3 when you look at layer 6, where we've got light regions of low expression, we've got dark regions of high expression in layer 6. It's really bringing something out of the data set now. Okay, so that's kind of goal 1 achieved. We've quantified cortex-wide gene expression in a layer-specific way. So the other question is how to sort of compare across brains, and to compare different brains, because they come out in slightly different shapes, different sizes. We need to sort of transform them so that they're on a common coordinate system. And to do that, we came up with this idea of putting landmarks on the data. So this image here shows these are the axis marks that I've already talked about, the needle alignment marks rather, and this orange point here is how you define that user-defined brain axis in the image. So you apply that mark on the image, and that says where the end of the brain axis is. But these blue marks are the global landmarks, and if you define 3 global landmarks in a set of brain slices for one brain, what you can end up with is 3. I've marked them here on a sort of 3D render of that brain. And when you then flatten that brain out, you end up with a triangle of locations. And so if you've got a second brain with the same set of global landmarks, which have been placed in sort of anatomically repeatable locations, you can then find a linear transformation to transform the second triangle so it matches the first triangle. And that means you can then apply that same linear transformation to all of the locations on the second brain, so that your 2 brains are effectively on the same coordinate system, as defined by the sort of the quality of your anatomical landmarks. And so this is kind of what you get. This is so the right. So ignore the kind of the mishmash of color stuff there. That's just three 3D surfaces all plotted in the same location. So that's not very useful. But what we've got here on the right here is the first brain. This is all layer 2/3 expression in 3 different vol brains. Because you can see that the individual slices are straight in this map. So this is the kind of template brain. And then these 2 other brains have had the transformation applied to them, so they've got different amounts of sort of twist to them. And then when you look at the resampled maps, you can see that we've got this region of high expression is repeated in the same location on all the three maps. So this gives us the opportunity then to take these three images, and we can do correlation analyses on the images, and we're confident they're in the same coordinate system. Okay, so that's kind of coordinate systems looked at. I wondered if you could apply this technique to other regions of the brain. I mean, the problem with the cortex flattening approach is that it's great if you're interested in the cortex, but not so great if you're interested in something else. So I thought, well, the hippocampus would be interesting because it's got this amazing spiral shape. So I prepared I took our mouse and vole data and I drew spiral hippocampus curves on all of the samples. And then these, you can imagine these being unwrapped and flattened out to get a sort of flat map of the hippocampus. And it looks like this. So we've got 3 vole brain here, 3 vole hippocampus maps, and 3 mouse hippocampus maps. Again, it's ID2 expression. And you can see that this one is the straight one. So all of the other ones have been transformed into the coordinate system of this vole brain. And we applied the correlation between species and across species, and found that there is strong correlation within the mouse brains, strong correlation within the vole brains, and very little correlation across the species in this case, and at this developmental time point. So you can, I was quite pleased with these unwrapped hippocampuses. They're quite fun. I mean, the key difference looking at them is the expression in this lower part of the maps where we've got much higher expression. Oh, I should mention that in this case, white is high expression in contrast to the other images. But we've got higher expression in the lower part of these mouse maps, which is largely absent in the vole hippocampus. You can also look at the expression as a function of depth. So if you take each of the sampling bins and you look at the expression down the bin, you can plot it out. I've done that for 6 locations across this brain slice. You can then even take all of those depth-based expressions and sort of put them back together again to give you the depth expression as a function of linear distance along the curve, which is what's plotted in this color plot. I compared the Allen developing mouse brain data with our technique, or rather I applied our technique to the Allen data. So I had to kind of reverse engineer the color expression. So these are the slices that you get from the Allen website have got this kind of 2-tone color scheme, and nobody really seemed to know what the kind of relationship was between the color and the actual expression level. But they did have they had an expression level map as well. So I took those together and kind of came up with a sort of reverse engineered scheme to get the signal out of the original images. And that's what's plotted in C here. So the white regions of the signal. But having done that, and produced, this is a mouse surface from the Allen brain map here, we got reasonably good correlation with our mouse data, so that when we flattened the maps out, we had a reasonable correlation in the regions where we had data for both maps. The reason they look so different in terms of their sort of shape is that the Allen data was sagittally sliced, and this slice set didn't cover a complete hemisphere, whereas ours was coronally sliced and did cover a full hemisphere. Okay, so you can try it out. You can try it out on an Apple Mac. I've done binary builds for the Apple Mac. They're available on the GitHub site. And there's a stalefish.dmg file that installs it. You'll end up with an icon that you can click. If you click it, you won't see much. You just get this white window which says "Welcome to Stalefish" and gives you some information. And the reason for that is that you normally have to run the program from the command line, and you have to give it the name of a text file, which is your configuration for the project. Okay, I avoided bringing in any kind of GUI toolkits into this program. I wanted to keep the code as small as possible, so it's got a very simple interface. You can also get it on Linux. I put it on the Snap store for Linux, which is a really great way of distributing applications on Linux, because it means that you can deliver it for any distribution. It connects directly to the GitHub, and every time I update the GitHub with a bug fix or a feature enhancement, the builds are automatic on the Snap store. And it also means that it shows up in the Ubuntu software app, which is nice. There's basically two programs that you get. One is called Stalefish, and that's the program that allows you to annotate the brain slices with your curves and your landmarks. The other program is SFView, which actually started out as just a quick hack to have a look at the 3D surfaces, but it ended up it does the linear transform stuff. So you load SFView up with multiple brains, and it then transforms all of the brains onto one. Okay, so two programs there. To create a project, you basically create this configuration file, which is in the JSON format. And it's a pretty simple file. This is a kind of minimal example I'm showing here. You would have more slices in your brain set, but here we've got 4 example brain slices, which we list in a little array. And we tell the program where their position along the kind of brain axis is. That's the X axis. And then there's a few other bits of information here that you give to the program. There's a supplement.pdf file in the GitHub which describes what those are, but they're things like pixels per millimeter to define, the scale of your images and so on. And then you run Stalefish with this JSON file as the single argument, and it launches the program. And you get a window looking like that. And it basically allows you to click points around the curve and click points for the landmarks. The most important key to know, because it's got an interface where you use key presses to change functions, is H for help. And if you press H, you will get an overlay of some text to tell you what the keys do. I've put arrows on here just to sort of highlight the ones that are most frequently used. So there's a bunch of functionality there. It's sort of a bit like going back to the Windows 3.1 days in a way, but that's the way it is. It works. So when you've annotated your slice setup, there's keys for switching between the slices in your brain set. When you're finished, you press W and it writes out an HDF5 file. HDF5 is a sort of standard binary format, which is good for storing data, because it means you can then open the data in other tools like Python or Octave, whatever you want. So having done that, you then open that H5 file in SFView. I've put a couple of example command lines that would do that. You can see that because I called my minimal example min.json, Stalefish has written out the H5 file and named it min.h5. And then I put in the path to my SFView program and launched the viewer. SFView does have some options. They're all command line options, and you can find out what they are with -? And then really, it's there for you to extend the analysis. The intention of this tool was to kind of capture the data, put it in an HDF5 file, and then allow the experimentalist to that data and do their own analyses. So the form of the data in the HDF5 file is all documented within the GitHub repository. There's a within that supplemental data file, and I think there's some readmes as well on the website. And there's also some examples of how you can open and read the data. There's a directory called reading/Python and reading/Octave where you can see how to do it. I'm sure we haven't done everything that you could do with the data that you can capture this way. So there we are. There's a methods preprint on this work, which has been carried out. We've been doing this for about 1.5 years, I'd say. That's the link to the GitHub, and by all means contact me via Twitter, or create issues on the GitHub webpage to seek help. So that's it. I'll just finish by saying thanks to our funders, the James S. McDonald Foundation. That's it. Thank you.
Thank you very much, Seb. So the question I wrote here on the chat on Zoom and also on YouTube that people could just ask questions during the talk. Now there is more time for questions, and I guess while people warm up, I can ask a couple of them myself if you don't mind.
Yeah.
And the first one, it's related to this last part where you're showing. So I actually have two questions. So you showed that you develop packages for Mac and Ubuntu, right? What about Windows? And the reason why I asked this is that from my experience, most biologists who are working with molecular stuff and so on, they're not heavy, Linux users, although they should be, but they're not heavy Linux users and so on.
Well, it's simply development time. So in principle, it should compile for Windows.
Right.
I just haven't got the time to do that. So that's why I haven't done it. So yeah, it's unfortunately, I just haven't got the time to develop it for Windows.
And even having now Windows having this Linux subsystem, would that make anything easier in that sense? To deploy it there? Or?
So yeah, I've played with that. The Stalefish program should work on Windows subsystem for Linux.
Okay.
The OpenGL part of it, the last time I tried running it, I was quite hopeful that it might work in the kind of more recent, like there's a version 2 of Windows subsystem for Linux. But unfortunately, the OpenGL stuff didn't yet just work out the box. That's another reason it didn't happen. I mean, I think I'd like to sort of gauge in, if I find that there's interest in the tool, it would be worth putting the time into getting it running on Windows for sure.
Yeah. And so the other question that I have is plugged directly into that. So people who are interested, how do you find, like how easy do they find of starting to use a tool that is not, I guess, what they're used of using in terms of being more of a command line thing and not having a user interface and so on?
Yeah. I mean, so it's only command line in the sense that that's how you have to launch it, right? It unfortunately you have to read the documentation.
Right.
So a lot of these, I mean, this is kind of interesting. A lot of these open source programs, people get funded on some project, like we've been on this McDonald Foundation project, and we've been funded for a few years, and we've built a tool, and I've seen lots of tools that kind of they get built, and people put a lot of effort into the user interface and making them really great, but then still nobody uses them. Right. So we've put only just enough development effort into this tool to make it do what we wanted it to do. We haven't invested time in making it user-friendly, because that probably would have doubled the amount of time that it would have taken to develop it. Yeah. And there's no guarantee that it may be that the neuroscience community is not interested in this kind of analysis. It may fall flat. You just don't know. So yeah. I mean, it will be interesting to see if people do pick it up once. The paper is under review, so we're hoping it will come out relatively soon, but we're having to take some more data to kind of beef up the results in the paper.
Right.
Yeah.
So I don't know if I missed this, but you mentioned this. You're saying that you're developing this new kind of analysis. What are people using right now when they're not using Stalefish? Basically, how do they compare these different brains, and how do they align these things and so on?
Yeah, it's pretty much either by kind of making the cartoon drawings and sort of estimating, or using whole mount stuff. where you just stain the entire brain without slicing it, and you just look at the surface expression. That, as far as I'm aware, is what most people are doing.
Okay, but then it's really, I mean, and I'm sorry if I missed this before, but then it's really great that you're basically managing from, because people have slides stored in the labs for years, right?
Yeah.
So you could actually, with this tool, people could actually tap into that and like do completely new analysis on data that they already
and
spend a lot of time to do, right?
Yeah, that hasn't escaped our notice. We're aware of that, and it actually there is work going on within Leah's lab on thinking about how to maybe curate sort of existing data sets so that we can we can get more information out of data that has been obtained at great expense and with the use of a lot of lab animals. So yeah.
Oh, that's interesting. That's cool. So I'd like to ask again our audience to see if anybody has any questions. And so we'll wait for a little bit. And in the meantime, yeah, I wanted to ask. So you were working with collaboration with this with the labs in the US. You have more or less an idea of the user base that you guys have right now? Is it just these labs that you're collaborating?
It's just our lab, yeah.
Okay, yeah. Or have you heard? I was going to ask, or have you heard from people that said, "Oh, I found this online," and
No. I mean, the Snap installation, the Snap store tells me that there's about 20 people who've got the tool installed across the world.
Okay.
So it's just a handful of people have like had a sort of exploratory look at it. We did, I think Mac gave a talk about it at the JB Johnson Club last year, and I think a few people talked to him about it at that point. I don't think we were quite in a position to sort of share the software at that time.
Right.
But I know that there was at least there were a few people who were actually had said, "Oh yeah, I could use this in my research." So there's time will tell, but there's certainly we've seen some interest in it.
Okay, so hopefully this talk and things that are open neuroscience will also get some more interested about this.
Yeah,
I don't see any questions on the chat here or on YouTube. I guess, yeah, I think I don't have any more questions. I don't know if there is anything else you'd like to share.
No. I think I've sort of covered what the tool does. So let's see if people will by all means contact me, at a later date when you've had a look at it.
Okay, great.
I'm always happy to answer queries.
Okay, and so the way to contact you is, as you put here on the lesson slides, is Twitter or creating an issue on GitHub.
Yeah, or just search me up on the University of Sheffield Department of Psychology website. You'll find me there, and if you prefer to send email. Okay, great. So yeah, so people are now aware on how they can contact you. Yeah. And so thank you very much. I think this basically concludes our session today. Thank you everybody that joined in the audience. I guess everybody's a little bit more shy today than usual, but thanks anyways for joining. And with this, we can close our transmission. Thank you. Okay, so we are not live.